Graded potentials and action potentials are two types of electrical signals that play a crucial role in the communication within and between neurons. Graded potentials are small changes in the electrical potential of a neuron's cell membrane. They can be either depolarizing or hyperpolarizing, meaning they can increase or decrease the likelihood of an action potential being generated. Graded potentials are typically generated in response to sensory input or synaptic activity, Mild, 1981. When a neuron receives a stimulus, such as pressure or heat, the sensory receptors convert that stimulus into a graded potential. Graded potentials are localized and can vary in strength depending on the intensity of the stimulus. If the graded potential reaches a certain threshold, it can trigger an action potential.
Action potentials, also known as nerve impulses, are brief and rapid changes in the electrical potential of a neuron's cell membrane. They are the means by which neurons transmit signals over long distances. Action potentials are all-or-nothing events, meaning they either occur fully or not at all. An action potential is initiated when the graded potential reaches a threshold level of depolarization. This threshold triggers the opening of voltage-gated ion channels, allowing an influx of positively charged ions, such as sodium ions, into the neuron. This influx of positive charge causes a rapid depolarization of the cell membrane, resulting in the rising phase of the action potential. Once the membrane potential reaches a peak, the voltage-gated sodium channels close, and voltage-gated potassium channels open. Potassium ions then flow out of the cell, leading to repolarization of the membrane potential. This repolarization phase brings the membrane potential back to its resting state, or even slightly hyperpolarized. After repolarization, the cell membrane goes through a short period called the refractory period, during which it is temporarily unresponsive to further stimulation. This refractory period ensures that action potentials propagate in one direction and allows for the proper timing and coordination of neuronal signaling. A refractory period refers to a brief period of time following a physiological or electrical event during which the system or cell is temporarily unresponsive to further stimulation. It is a characteristic feature of various biological processes, including nerve cell signaling, muscle contraction, and cardiac activity. In the context of nerve cells, the refractory period is observed after an action potential, which is an electrical impulse that travels along the neuron. Once an action potential is generated, there is a short period during which the neuron is unable to generate another action potential, regardless of the strength of the stimulus. This refractory period ensures that action potentials propagate in only one direction and helps maintain the integrity and efficiency of nerve signaling. The refractory period can be divided into two phases: the absolute refractory period and the relative refractory period. During the absolute refractory period, which is the initial phase, the cell is completely unresponsive to any additional stimuli. This period is caused by the inactivation of voltage-gated sodium channels, which are responsible for initiating and propagating the action potential. As long as these channels are inactivated, no new action potential can be generated. A neuron, also known as a nerve cell, is the fundamental building block of the nervous system. It consists of three main parts: the cell body, dendrites, and an axon. The cell body, also called the soma, contains the nucleus and other essential cellular components. It serves as the control center of the neuron. Dendrites are branching extensions that receive signals from other neurons or sensory receptors. They act as the input region of the neuron, collecting information from the external environment or other neurons. The axon is a long fiber that extends from the cell body and carries signals away from the neuron. It can be covered by a myelin sheath, which helps to insulate and speed up the transmission of electrical signals. At the end of the axon, there are terminal branches that form synapses, which are specialized junctions with other neurons or target cells. These synapses allow for communication between neurons by transmitting signals chemically through neurotransmitters. A synapse is a specialized junction between two neurons or a neuron and a target cell, where communication occurs. It consists of three main components: the presynaptic terminal, the synaptic cleft, and the postsynaptic terminal. The presynaptic terminal is located at the end of the axon of the sending neuron. It contains vesicles filled with neurotransmitters, which are chemical messengers that transmit signals between neurons. When an electrical signal, known as an action potential, reaches the presynaptic terminal, it triggers the release of neurotransmitters into the synaptic cleft. The synaptic cleft is a small gap that separates the presynaptic terminal from the postsynaptic terminal, Savchenko, 2007. It acts as a physical barrier between the two neurons, ensuring that the signals are transmitted in a specific and controlled manner. The postsynaptic terminal is the specialized region of the receiving neuron or target cell that contains receptors for neurotransmitters. When neurotransmitters are released into the synaptic cleft, they bind to these receptors, initiating a response in the postsynaptic neuron. This response can be excitatory, meaning it increases the likelihood of the receiving neuron firing an action potential, or inhibitory, meaning it decreases the likelihood of firing an action potential. IPSP stands for inhibitory postsynaptic potential. It is a type of electrical signal that occurs in the postsynaptic neuron or target cell when it receives inhibitory neurotransmitters from the presynaptic neuron. During an IPSP, inhibitory neurotransmitters, such as gamma-aminobutyric acid, GABA, or glycine, bind to specific receptors on the postsynaptic membrane. This binding triggers the opening of ion channels, usually chloride channels, which allows negatively charged chloride ions to enter the cell. This means that the membrane potential becomes more negative, making it less likely for the postsynaptic neuron to generate an action potential. In other words, IPSPs inhibit or decrease the excitability of the postsynaptic neuron, reducing the chances of it firing an action potential and transmitting signals to other neurons or target cells. EPSP stands for excitatory postsynaptic potential. It is a type of electrical signal that occurs in the postsynaptic neuron or target cell when it receives excitatory neurotransmitters from the presynaptic neuron. During an EPSP, excitatory neurotransmitters, such as glutamate, bind to specific receptors on the postsynaptic membrane, triggering the opening of ion channels, usually sodium channels. This depolarization brings the postsynaptic neuron closer to its threshold for firing an action potential. In other words, EPSPs increase the excitability of the postsynaptic neuron, making it more likely to generate an action potential and transmit signals to other neurons or target cells. EPSPs play a crucial role in the communication and integration of signals in the nervous system. When multiple EPSPs occur simultaneously and are strong enough to bring the postsynaptic neuron to its threshold, they can trigger the generation of an action potential, allowing for the transmission of information between neurons.